Week 5 Ecology Research Project Presentation, Week 3 Focus, T. Blatnick. Biodiversity loss is by far the most concerning issue we face today, Hansky, 2011. In this experiment, 2 areas were analyzed. Both are surrounded by urban sprawl and heavily used by humans and dogs for recreation. However, one area has been restricted from human access for almost a year and dogs for decades. I wanted to see if this had any impact on the species richness of these 2 areas. The remarkable thing is that both sites share the same general space, the same resources, they share the same weather, waterfall, from rainy seasons. However, it's not clear if one site has a better growing potential because it's less disturbed condition, or if there's any difference in the amount of resources that affect the population density of each sites. Both of these sites are kind of nestled in this vast urban sprawl, and so they are extremely isolated. They are like little islands. Most species, with that said, need some sort of outside influence in order to not drown out their resources or become genetically inept. But they also need a stable environment in which to thrive. Whitlock, M.C., Barton, N.H., 1997. Research question: which area will have greater aquatic plant species richness and aquatic plant diversity? Hypothesis: I hypothesize that less human disturbance will positively affect aquatic plant species richness and diversity. Prediction: I expect to see greater diversity of aquatic plants and more comprehensive species richness in Site 1, the wildlife sanctuary. So this is my study area. It is Crown Hill Park, and it's located in Jefferson County, Colorado. Up here, this is Kestrel Pond, and this is the wildlife sanctuary in the northwest corner here. Crown Hill Lake is down here. This is my study site 2, and the remainder of Crown Hill Park. If you'll notice here, these 2 red dots, that's my transect line for Kestrel Pond, site 1, and then down here, these 2 blue dots, that is my transect line for site 2, and I walked along the shoreline here. So site 1, the sanctuary essentially makes up a quarter of the park, give or take, right? It would be considered marshland. It is also a protected site, so it has not been visited by humans other than permit holders, which I did obtain a permit for this project, or park rangers for about a year now. Normally it's only closed down for nesting season, but we did see excess rainfall. One of the wood bridges was washed out, and so no outside human influence has been in this park for about a year. There's no motorized vehicles, no dogs, no horses, just human foot traffic, specifically on the trails when it is open. The remaining park is frequented by dog walkers, joggers, families, school trips. It is packed. It's a very bustling place. There are a lot of fisher people that come in and fish year round, and so there's a lot of erosion along the shorelines. Oh, as well as Olinger Cemetery, which is adjacent to the park, it does have water rights to Crown Hill Lake, but not Kestrel Pond. And so there is periodic draining of Crown Hill Lake in order to water the lawns at the cemetery. So the variable that differs between these 2 study sites is human influence by way of recreation, essentially. Also by the water rates, right? We talked about that briefly. The thing about it is that the addition of recreational fishing on Crown Hill Lake shoreline causes a huge amount of erosion around the lake as a whole. There's a very long-standing history of this specific site. Like, the whole thing is supposed to be protected, but really only the sanctuary is. There's a huge amount of human impact on Crown Hill Park outside of that sanctuary. So the aquatic plant Typholatifolia, or better known as broadleaf cattail, is a problematic plant in both areas of the park because it can grow very fast even in harsh environments. Stevens, 2001. To assess its impact, measurements of Typha latifolia in my week 4 were done. I just wanted to essentially see, like, what's the potential of seed distribution? How much bigger are these plants? And I noticed that there was a difference by about 20%. So generally speaking, the plants in Crown Hill Lake Site 2 were about 20% larger than the plants, the Typha latifolia in Site 1. This is just an interesting side note that I noticed. There was another study done by Grace JV and Wetzel RG back in 1982 where there was no sort of positive correlation between the ramette size of Typha latifolia versus their ability to choke out other species. But I didn't look at ramette size necessarily. I looked at the size of the plant stalks and the size of the plant seed pods. And so that may just be an interesting side note. Who knows? So why is this a good area in which to test this hypothesis? So we've already talked about this pretty clearly, but Crown Hill Park is nestled in an urban environment. It's surrounded by businesses, schools, churches, homes. It's pretty isolated as far as a wild space within a very urban environment. It has a rather robust diversity profile given its location, but in this space there are these two separate and distinct areas, right? And the thing that's interesting about studying aquatic plants specifically in this area is that there's two separate bodies of water, one that sees zero to very little human impact and another one that sees a huge amount of human impact on fishing and recreation and water use. They're very different but also very, very similar. As far as aquatic plants, these organisms are very interesting because they live in a very specific environment, right? I could have studied grasses or trees or shrubs or any number of things, right? But the thing about it is that these plants have to live next to or into water. And so there is almost a very like laboratory-like setting. Here's this one body of water, here's this other body of water. Let's study the aquatic plants in both. So my sampling methods, I walked a transect line like I showed you on the map, and I did go about 5 feet into the water and then about 10 feet off of that shoreline and identified specific individuals. And then once I did that, I did have to do some estimations for some species. So for duckweed, for example, it's a very, very, very tiny aquatic plant. And so I would take a smaller section, count all of the individuals in there, and then essentially multiply that over the full area where this plant was present. And I did do the same thing with cattails. Very interesting thing about cattails, they do grow by rhizome, and this is their predominant reproductive method that they use. And essentially what that means is that they have one individual, and that individual will send off a root and then shoot up a genetically identical individual. So they're not necessarily separate plants, right? They're all the same. But what I did instead was counted the individual stalk as one plant necessarily, and then multiplied that over a larger space. So here are the results of my Site 1. You can see this is a rounded number here. I'm not super pleased about that, but once again, I don't have the ability to count literal millions of duckweed and thousands upon thousands of cattails. I did see some smartweed here. I also saw asparagus, one of my favorite plants, and then also another one of my favorite plants, wapato, which has been utilized by indigenous peoples for millennia. Take a note here at these numbers. So my Shannon index is almost 60%. My evenness is closer to 40. We have 5 species here. Site 2, I was only able to find 3 aquatic plant species. And my Shannon index is much lower, and so is my evenness here. So with that said, my hypothesis is supported by my data. But that doesn't necessarily mean that it's definitely true. There may be species that I missed. Maybe there's a surplus of duckweed in the spring or the summer. It's hard to say, right? I took this data during the fall. An alternative hypothesis is that, there's also more birds in the sanctuary, and birds allow for seed dispersal as well as providing fertilization. Maybe that's a factor. Some of the ecological factors that may have influenced my results are potentially the size of the bodies of water. Kestrel Pond is much smaller than Crown Hill Lake, even if Crown Hill Lake is experiencing, periodic drainage, right? And erosion due to fishing. So some of the logistical or non-ecological factors that may have influenced my results are still those, impacts by humans and dogs. If I were to repeat this work, I would repeat the process, but for different seasons. And in addition to that, I would build on this by looking at more comprehensively the whole of Crown Hill shoreline and Kestrel Pond shoreline. I wouldn't have taken just a small area. I would have done both sites as a whole, and maybe potentially look at, not only the portions of both sites that were studied, but, maybe try to compare in size too. I think that my results raise more questions than answers. Although my hypothesis was supported, I think that there's other factors that could explain these results. The most significant finding is that there is a negative impact on the biodiversity and species richness of Site 2. It's arguably caused by human activities, right? But there are two studies, Habitat Loss, the Dynamics of Biodiversity and Perspective on Conservation, Hansky 2011, and the Effective Size of a Subdivided Population, Whitlock, MC, Barton, NH, 1997, that showed that one aquatic plant species, Typho latifolia, which we found in both sites, can impact other aquatic plants if the water levels change or if the habitat quality is affected by an external source, respectively. So this does suggest a strong correlation between human impact and diversity, and that maybe this particular aquatic plant is taking an advantageous hold on the situation. However, further research shows that the species richness of an aquatic plant species has an impact on the biodiversity of an area as a whole. So, yes, this is happening. Maybe it's human impact that's causing it, but the biodiversity of a whole of this area is affected just by looking at the aquatic plant species. With that said, some aquatic plants, it's been noted in several studies already that they need better management, cattails specifically, especially in areas with human impact. Thank you so much for looking at my presentation. These are my references, and have a good night.